Okay, well, good afternoon, everybody, and welcome to this last of the 5 MERCES webinars that we've got. MERCES, it's a Horizon 2020 project for blue growth, and it stands for Marine Ecosystem Restoration in Changing European Seas. It started in June 2016 and continues through to the end of May this year. And we will probably have lots of different deliverables that will be published online during these final few months. And if you'd like to keep an eye on those, I've put the website here for you, and you can just log into that, and the various deliverables, once they've been agreed, will be made available for people to see. So welcome anyway to this. We're very pleased to be working with Tanya Bryan and Rob Barnes at the GRID-Arendal in Norway to bring this webinar to you. And I'm also very grateful to Eva Ramirez-Lodra for being my co-convener and helping me with a lot of the decisions and her wise advice. And also here where I am at the National Oceanography Centre in Southampton, thank very much Andrew Gates, who is also hosting me in his office to make sure we have a secure connection for this webinar. To fulfill the aims of blue growth, MERCES has been attempting to bring industry and policymakers and scientists all together with a common cause in marine ecosystem restoration. I think, as we will see in our talks today, it's by combining all these different skills and interests that we actually get some progress in marine ecosystem restoration. And the project itself from MERCES, you will find, goes all the way from the coastal zone and many different types of coastal habitats, including seagrasses and corals, but also moving into the deep sea, so taking up the restoration of deep sea corals that may have been impacted by fishing, and also looking at issues that might now relate to the possible mining of minerals in the deep ocean and the restoration of ecosystems that may be required from that. So if you wish to see anything to do with MERCES, do go to the website. And we hope that through our newsletters and the case studies, and also the archive webinars that you can find on the MERCES website, that you'll see the range of activities that are now possible. So next slide, please. So today, I'm very pleased to be able to introduce to you 2 speakers, but they are backed in both cases by formidable teams, as no doubt they will let you know. So we will start off first of all with Jesper Elzinga of Van Oord Dredging and Marine Contractors in the Netherlands, talking about some new work that was reported in Frontiers of Marine Science last year on the sort of recovery of reefs, of coral reefs at the industrial scale. We're trying to look at ways that we can actually make a difference at a bigger scale. And this seemed to me a particularly novel and good approach. And then we will follow that up with some work that has been going on within the MERCES project, both in shallow water and in deep water. And Joachim Garibou will be the presenter, but he will also show that obviously Cristina Linares at the University of Barcelona, and also Marina Carrero Silva at the University of the Azores, and their teams have also contributed a lot to this work. So that's what will happen. Now, during the presentations, you are able to pose questions using the question and answer box, which you can find on your screen, either at the bottom or the top. And if you type your questions in there, we will have a moderated discussion at the end of the webinar. So Each speaker will take about 20 minutes, and then we will have about 20 minutes for the question and answer session. And I will do my best to answer all your questions or pose them to the speakers and to have them answered before we close. If not, then I will try and arrange for a way that the answers might be supplied in written way and posted on the MERCES website. So please use that Q&A box. It helps me as a moderator to see the questions as you pose them. So don't wait to the end of all the talks before you pose your questions. Do populate that Q&A box as you think of a question and during the talks. And that way I can be better prepared, and also the speakers are able to see those questions. So that's the way we want to go. There is also a chat box, but that's really for technical questions like, can you hear me or can I be heard? And so I'd much rather, if you could avoid using the chat box, just use the Q&A box. So without further ado, I'd like to introduce our first speaker, Jesper Elzinga of Van Oord, to make his presentation. Over to you, Jesper.
Okay, I'm just trying to get into the presentation mode. There we go. Okay, can you hear me okay, Dave? Dave, can you hear me okay?
We can hear you, Jesper.
Okay, thank you very much. Thanks for the introduction. First of all, my name is Jesper Elzinga. I work for a company called Van Oord Dredging and Marine Contractors. I joined Van Oord in 2013 with a background in aquatic ecology, and then worked there ever since as an environmental engineer. And David's invited me to discuss the recent Recruit project we executed together with a few partners. And Recruit stands for the recovery of reefs using industrial scale techniques for slick harvesting and release. I'm not sure how familiar everyone is with coral spawning and the general state of coral reefs. Because of this, I have tried to keep a nice mix between hardcore science and the fundamentals of the techniques we aim to apply. However, if there's questions, please feel free to ask them in the comment box, or you can email me, and I'll be very open to answer them. So first of all, I'd like to discuss the partners with who we conducted the research. So first of all, I work for Van Oord. We're a marine contractor. We have 150 years of experience in marine infrastructure. This includes, for example, dredging, reclamation, construction of ports. So one of the Palm Islands was actually constructed by us. We also work with the building of offshore wind parks, subsea rock installation, pipeline installations for different purposes. Another one of the partners that we worked with was CSIRO, the Commonwealth Scientific Institute, the Common Scientific and Industrial Research Organisation. They're an Australian organisation, and they have a mission to create benefit for Australia through impactful science and innovation. And we also work together with the University of Delft, Technical University, well known within the Netherlands and also worldwide. And they helped us with some of the hydraulic components of the research. Us three partners, we teamed up together for the Advanced Queensland program. It was a program boosted in 2018. And the purpose of that was to boost coral abundance on the Great Barrier Reef. And we came together with these partners. We had a goal, and the goal was to upscale the current coral restoration efforts by 2 or 3 scales of magnitude. And one of the nice things, which actually ties in quite well with what Dave was saying, is that we have different parties with very different expertise, yet we can bring them together and really complement each other's expertise. Here I have a quick sketch of the work method, which we proposed and tested to a scale for the research we conducted. And this is where some of you may think, well, what I see here is relatively fundamental, and for some people it might be quite new. However, coral typically spawns once per year in a mass event. That is, with broadcast species, they release bundles which contain eggs and sperm. These are then released, they rise up to the water level where they open up, fertilization takes place, then over the course of around about a week, they travel and they develop further, and then they reach settlement competency, at which point they swim down towards the bottom, attach themselves, metamorphose into a coral polyp, and then for Acroporus species, which we've looked at before, it takes approximately 4 to 5 years before they reach an age where they can reproduce themselves. The technique that we've proposed is when this event is occurring, we show up with a vessel, we look if we can find the slicks. So when the spawning's occurred, it forms into a slick. We see, can we concentrate this slick? Can we then harvest a part of this slick from the reef and bring it into one of our vessels? These vessels are known as trailer suction hopper dredgers, in this case of the model, which have a hopper or a barge-like structure within it where you can contain a lot of water or sediments for different purposes. From this point, we would sail from a reef which is producing, which is healthy, and sail to a reef which may be degraded. And then at that reef, we would like to release the competent larvae. So it means the larvae are able to settle onto a reef, with the goal of then rehabilitating a stretch of reef which has been impacted. We've modeled all of this in the paper together with Chris Staropoulos, myself, Remington Hofstede, a colleague of mine, Mark van Koningsveld, who works for the TU Delft, as well as for Van Oort, and Russ Babcock. The cool thing is, we actually got the opportunity to test some of our modeling. So it's nice to have a model, as it allows you to have some form of a reflection of reality. However, there was a lot of uncertainties. And one of these uncertainties, for example, was, can we actually find mass spawn slicks out in the Great Barrier Reef? And once we found these slicks, what do we actually do? Can we concentrate these slicks, or is that impossible? Another thing which is very exciting to me, I thought, look, what if we find them and we actually are able to pump these into our vessel? Will they survive the pumping? It's a very critical period of time during which we collect these coral larvae and gametes as well, of course. Do they actually survive this process? And then once we have them on the vessel, are we able to keep them alive in the vessel? So the inside of our barges or hoppers is typically made from steel. Would that lead to certain water qualities which are not desirable for coral larvae while they're developing, or are they okay with this? And then last of all, what was really important was to find, can we actually produce larvae which are able to, which are settlement competent, which means that if given the opportunity, they're able to settle to a reef and metamorphose and continue, hopefully, to maturation. So to test these different uncertainties, we ran a pilot project on the Great Barrier Reef. We did this in November of 2008 during the mass spawning events near Heron Island. And to test the uncertainties which we faced, we used a barge. And on that barge, we had set up 12 different water tanks, some of which were plastic, others were made from steel to reflect the inside of our vessels. Upon this barge, we built an aquaculture system, and we had 2 different pumps which we test to see, hey, look, do these larvae actually survive if we pull them through pump? This is further described also in the paper which we wrote, very similar authors, and that's actually the paper which David referred to in his introduction. What I'll do now is I'll run through the different uncertainties which I've just discussed, and the ways that we investigated these and the results which we retrieved from that. So we'll quickly go into the results section already. So the first question we had, of course, is can we actually find dense slicks? And one thing I really like about this is we first did hydrodynamic modeling to predict, hey, can we predict where these slicks are? And then to verify, we had a helicopter. I'll show you the footage right now. So what we can see is the slicks were actually where we modeled they would be, which is very, very exciting that this is predictable. Unfortunately, this isn't my footage in the helicopter. I very much would have liked to ride the helicopter, but as the second youngest guy in the project, I was informed there was a certain level of seniority required to ride the helicopter. So I'm hopeful that in future projects I have a different task. And the fun thing is actually on the right-hand side of the image, you now see the tug that we were working with in the background. So we are really close to this slick and we are ready to go towards and see if we can test our further research questions. Second research question we had was, are we able to concentrate slicks to a point where we can harvest these? So the main means for us to investigate this is that we first off had to find slicks. Well, I'm quite happy to say that we found them. Secondly, we would send a pilot vessel with an oil burn, which is seen on the right-hand side. So we have a little pilot vessel and it's got a screen behind it. The purpose of that screen is to essentially capture the slick so that we can come over with the mother vessel, if you will, and be able to pump this up. The results were quite encouraging. So we had high densities of slick within the oil burn. And in fact, we actually had certain samples which had a concentration of 170,000 embryos per litre, which is far greater than what we expected. And that's encouraging to see because then we can actually hopefully harvest quite large amounts of embryos from a location, whilst not impacting the population which is there at the moment. And then have more in the beginning of the process, which then means that we are able to bring back more later on onto the actual target locations. Another thing which I've quite enjoyed about this is that we were able to use technologies from different fields and integrate them to our purpose. So these screens are usually used to contain oil around construction sites. And we can use that same technology and learn from these practices to also contain different materials. Then one of the research uncertainties that we had is, which I in particular was quite skeptical of, are we able to get larvae and gametes through a pump without, well, surviving larvae and gametes through a pump? So the means that we investigated this is we did a literature review on a lot of different pumps and had a multi-criteria analysis together with the TU Delft and also experts within Van Oord to really investigate, hey, which pumps do we find most feasible for the purpose and which pumps do we expect provide low impact to the water within them and therefore hopefully the lowest impact to the actual larvae that we're pumping. We came to a short list of 3 potential pumps. So we had a hydrocell pump, which is essentially a screw within a pipe, which pushes the water further away as opposed to accelerating as such. We also had a diaphragm pump, also known as a membrane pump as a simile. And we had a centrifugal pump, which we tested mainly because the vessels that we use currently have centrifugal pumps. And if we are able to use this same technology, that'd be very beneficial. We tested this in lab conditions. And fun thing is we didn't actually have coral embryos available to us because spawning only typically occurs once a year. So as a proxy, we used small berries, for example. We also used hydrogel balls, to see, okay, look, are these impacted by the pumping process, and can we extrapolate this to coral embryos? The interesting thing here is that we saw a very low intact recovery rates on the centrifugal pump, and therefore we decided, look, this centrifugal pump really provides a lot of undesired pressure on the material that we're pumping, and therefore we decided, okay, we'll use a hydrostyle and a diaphragm pump to test within the field. Then we moved to the field. We found the slicks, we concentrated the slicks, we deployed our pumping mechanism, and we tested this with 2 different pump types. So yeah, we used both of which, and to my surprise, the survival rate was actually quite high. The survival rate was roughly 120%, which of course seems to be impossible, but the way that that worked is that we had some fragmentation during the pumping process. And we saw that these fragmented larvae actually developed further through the full cycle to settlement competency. So you have some form of clonal reproduction here as well, which is something that occurs out in the ocean as well, when you have wave impacts which break apart small bits of coral gametes. And if this is not too severe, then you have 2 gametes essentially continuing on. They're just slightly smaller than what they usually would be. Once we had the larvae on board, one of the next uncertainties that we faced in our model was, okay, do we need to rear these in plastic tanks, or can we do this in steel tanks? So essentially the question we're trying to answer here is, if we use commercial vessels, would we need to line the inside of these vessels with plastic, or can we work with the general steel setup that we had? The results here is that we had very comparable survival levels between the plastic and the steel tanks for a period of 100 hours. The reason we tested 100 hours is because after that period of time, we saw the first settlement behavior starting to kick in, which meant that we essentially reached the next step of our uncertainties. One thing that you do notice is you notice a reduction of survival over time. This looks somewhat alarming if you don't know the context, but this is actually something that you see out in nature as well. And if you look at the numbers in our model, these numbers are actually very similar to what we would have expected. And also, if we work with more controlled aquaculture environments, such as what we do in our reef guard project, for example, we also find fairly similar survival rates. Another question which we wanted to answer is, okay, once we've developed these larvae to a certain point, did they actually reach the essential points of settlement competency? And the way that we investigated that is we essentially had to execute the entire cycle of what we had monitored, of what we had modeled, sorry. So we had to pump and rear larvae in the plastic and steel containment tanks which we had. And we did that until we saw signs of competency. When we saw these signs of competency, we started introducing substrate into these tanks to try and notice, okay, do they actually successfully settle and connect themselves to this substrate? And this is an essential step in our potential work methodology, because once you release these larvae onto a reef, you want to make sure, okay, these larvae can actually attach themselves and they can grow up and then grow to maturation and then continue the spawning cycle again in a number of years. We found that settlements occurred in both the plastic and the steel tanks. One difference that we found is that the settlement was substantially higher in the plastic tanks. However, we suspect that is due to a slightly higher water temperature, which was in the steel tanks. So we found that over time, the steel tanks started getting a little bit warmer than the plastic tanks we had. So then I'd like to bring you guys back to the overall picture of what we were trying to do. So what we tested was on a certain scale with the intention to eventually upscale to a larger scale. And to do this, we wanted to test a number of uncertainties. So one of the things that we were uncertain about is, okay, can we find dense slicks? place a little tick on the screen because I'm happy to say we were able to find these. The second question was, okay, can we concentrate these slicks using common technologies that we know from marine infrastructure, essentially? We could also do this, so we were able to get quite dense slicks towards our pumping system. Next step was, can we pump these larvae onto our vessel without causing excessive harm of the population which we're trying to harvest? We were able to do so. Next step then was, okay, can we allow these larvae to survive within the relatively basic aquaculture setups that we had? So usually when we work with our reef guard project, we have a mobile aquaculture laboratory which we're able to move all over the world. And we can actually control the water temperature. We can control the quality of the water a lot better. So these conditions were quite coarse compared to what we usually would work with. We found we were able to get them to survive this stage in a similar fashion to what we usually see at our reef guard projects. And then last of all, we wanted to see, okay, can these larvae reach settlement competency? I'm happy to say we could actually get them to the point where they could settle onto a reef. One thing which we would like to conduct in the future is we'd like to actually release these larvae onto a target site which is potentially degraded, and then we can notice, okay, do these larvae actually settle here, and are they able to rehabilitate a select location which we are targeting? Yeah, I mean, that would really be exciting if we can take it to that extra step. Just wanted to run through a couple of potential applications that we see for further incorporation of this methodology. So the applications for this is we could translocate larvae from healthy to damaged reefs, and hereby facilitating rehabilitation. We could also facilitate more ecological connectivity between otherwise isolated populations, which is something that you see in the Caribbean, for example, that you have very isolated populations which don't typically manage to breed together. We could also translocate heat-tolerant corals from hotter areas to areas which are at risk of heat stress in the future. So essentially move water from a slightly warmer region to a slightly cooler region. We could also do heat hardening of the larvae during the translocation. So we could work with higher water temperatures on the vessel, making sure that the larvae which survive are more suitable for warmer water temperatures. There's also a potential to work with large-scale introduction of heat-tolerant cymbidium. Just a footnote, I mean, these things are all potential appliances of the technology. That doesn't necessarily mean that all of this is something that we are actively pursuing. The advantages that we see within this methodology is that there is no or negligible damage to the existing reef. So if you look at the massive spore slicks, which we saw on from the helicopter, also the ones that we see on the right-hand side of the screen, the amount that we can harvest at this stage are a very, very small subset of what's actually going on that reef, meaning that we have essentially no impact on that location. And another advantage is that we can essentially harvest the entire species assemblage. So a lot of the restoration work, which has previously been covered, covers a certain amount of target species or keystone species. Whereas with this, you really get the full complement of what is spawned on this event, and move it to an area which potentially has been degraded, and therefore introduce the whole species complement back at that area. As long as you do realize that we only move the broadcast species with this, of course, and not the breeding species. That concludes my presentation. I hope it's been informative for you guys, and I didn't get too far into the science or too fundamental about it. I'm sure I'll see some questions pop up, which I'll answer. There's also my email address, which you can reach me on if need be. And I've inserted a Vimeo link to our Van Oord Vimeo, which shows essentially a movie of the process and field images of what we've conducted for this recruit project. Thank you.
Okay. Well, thank you, Jesper, very much. And I'm sorry about the hiatus at the start. We were just changing screens and the unmute button disappeared.
Oh, right.
There we go. Now, while we change through now to Joachim's talk, I'm afraid that perhaps next time we won't have a helicopter ride, because no doubt costs will come in and people will start using drones instead. So I think you've had a chance on that one. You've got a lot of questions coming through already, which you can have a look at. And in fact, I think you answered a few maybe in subsequent parts of your talk. And also you indicated the future work you're going to be doing, which maybe also addresses some of the questions. But there are many useful questions there. And thank you for those that are participating that way, because it'll lead to a very good discussion in a moment. But before we go to the discussion, I'd like to introduce our other speaker, Joachim Garabou from CSIC in Barcelona, and he will tell you about work that's been going on in the MERCES project in both shallow and deep environments. Over to you, Joachim.
Okay, thank you very much, David, for the invitation to do this webinar. My name is, as David said, Joachim Garabou. I'm a senior scientist at the Institute of Marine Science here in Barcelona. And this institute, it belongs to the Spanish Research Council, which is one of the main research institutions in Spain. And my background is I'm a marine biologist, and I work for my entire career in marine conservation and marine protected areas. So restoration is a tool that we can use to support conservation. And within MERCES, my role was the co-coordinator of one of the work packages, work package 3 about rocky benthic ecosystems, but I've been in contact with people working in deeper waters in the continental shelf and in the deep sea. So in this webinar, I'll try to summarize what are the protocols that we have been using within MERCES. And this is the list of people that that has contributed in somehow to this to develop these protocols, and there are many others that I couldn't put here. But I mean, the point is that it's behind these protocols and these efforts, there are a lot of people working for. So the first thing, since we were in the tropical seas, which was really warm waters and crystal clear in some places, and I just want to give you an overview on what are the corals in the temperate and cold water waters, and this kind of the environments that we have where these temperate corals are developing. And the main differences that I want to highlight here is that when we talk about tropical corals, usually we are in a really light, with a lot of light environments, while in temperate and cold water corals there is that dim light habitats or no light at all habitat, right? So we don't have in these corals this endodymium or other photosynthetic symbionts, and the corals, they show a slower dynamics. The growth rates are much slower than in the case of tropical corals. Okay, but they play the similar ecological role as a habitat forming species, and in this sense, these are the habitats where we have been working. So we have I don't know if you see my pointer here, but we work in the coast, in the continental shelf, and then in the seamounts in the Atlantic, in the Azores Islands, and those in the coastal and continental shelf. We were working in the northwestern Mediterranean. So this role as a habitat forming species, they provide some key marine ecosystems, ecosystem services, I would say, like food provision by fishing, cultural services, recreational diving and tourist attractions, material provision as some compounds that can be used in the pharmaceutical companies or other materials that we can use for jewelry, for instance, for the red coral, and regulation because they can play a role as well as carbon sinks and cleaning the waters, etc. But as you know, we have these ecosystem services are at risk because we are in this trend of degradation of marine habitats and the ocean in general. And I'm just going to show you some two examples on what are the drivers of this degradation. For sure, you know, but it's good to remind, to recall this. And these are the ghost fishing nets, for instance, which is telling us that the fishing activities are playing, they can destroy the habitats where the corals develop. And more recently, during the last 2 decades in the temperate seas, we are witnessing for the first time mass mortality events which are linked to the climate change. Specifically, we have been working in the case of the Mediterranean, and 20 years ago we were the witness of the first large scale event, mass mortality event, encompassing more than 1,000 kilometers. But since then we have been witnessing different mass mortality events almost every year, and they are affected already more than 90 species from different kind of groups of species, from algae to corals, but as well as sponges, bryozoans, ascidians, etc. And most of those events have been linked, or they were concomitant with the occurrence of marine heat waves in the areas. Okay, so climate change is a main driver, actually a main driver of degradation in the Mediterranean, and as well in the tropical seas, as we are witnessing right now at this very moment at the Great Barrier Reef with the new onsetting new bleaching event. So the corals that we are dealing with, they are really highly vulnerable to the disturbances. I'm just going to give you some key information on why they are so vulnerable. And this information that I'm going to provide you, they come from long-term monitoring exercises or studies that we have been doing. So we go to the sea and we measure the different corals. Like here in this red gorgonian, and we use different techniques like sclerochlorology technique, that if this a cut of a red coral colony, so we can see the different growth rings, and then we can count the number of growth rings, and we validated that this each growth rings was developed on annual basis, and then we can infer the age, and we can calculate as well the growth rates. Okay, so for instance, in this case, we were comparing the older one of the common before the fishing, this kind of colonies, red coral colonies, were common, and they can have more than 200 years, but now we have just small size colonies. Okay, we have been conducting as well genetic studies, population genetic studies, to know the connectivity between populations and how they can recover from. I mean, if there are some populations that they can send the larvae to other affected populations to recover. Okay, so we have been developing this in many species. And as a summary of this vulnerability is that these species, when they suffer damages, the growth rates, they are really low, from 1 millimeter to 1 centimeter per year, more or less than this. They are really long-lived species. Most of them, they can have more than 100 years old. So if they are affected, you need at least the same time to recover the populations. And they have a limited dispersal, so you have to act at really the management or conservation actions. They can deal with local actions, and they show as well low recruitment rates. So this is really bad for them to come back after a degradation, right? So this is why restoration can support the resilience of these communities. And this is why we were involved in the MERCES project, and we developed some restoration protocols that you can find here. I put the names of the deliverables if you want to go to the web page of MERCES project and look for this. And as well, I'll mention it that I put the references of some of the works that where you can find in the information. Okay, these are the main species on which we have been working in the coastal, in the continental shelf, and the seamounts habitats. Just to give you a view on the kind of the species that we are working with. And in the coastal areas, we have been working with using scuba diving, so making transplants mainly. All the techniques that I'm going to show you today is transplants, because they were the main tool that we use. So here the transplants were done by scuba divers, and in the continental shelf and the seamounts, it's a little bit more complicated. There are some structures that they are placed in the bottom, and then you can make surveys with the ROVs and the submarines, and we will have a closer look afterwards. So I'm going to go from the bottom of the sea up to the coast, and this is the example of the seamounts in Azores. So this is the MERCES provided as well the opportunity to show how collaboration among stakeholders can take place for the restoration projects, and this is one of the cases. So the bycatch corals they brought to the lab. They kept them into the lab, and they saw that the survival was quite good. And then they placed it in these artificial structures, and they placed them into the bottom, and then they were checking the survival and other indicators on the status of these corals down into the seamounts. Okay, so depending on the species, the survival rate was quite low, 15%, but for some others, the survival rate reached up to 100% after 2 years. So it was quite good results in this sense. So if we move to the continental shelf now, again, here they were the people working for in this subject, in these restoration protocols. They were established a collaboration with fishermen in the Catalan coast. So they took these bycatch colonies, and then they used this kind of artificial structures to place them and fix them in these structures, and then put them up to 80 meters depth. Then they go with a rope, and they were checking the survival of these colonies, and they could quantify that a really good survival rate. About 90% of the colonies, they were there after 1 year of that they placed this structure in the bottom. But the people working in this on the continental shelf, they developed another interesting restoration protocol that they used. These are the bycatch coral, and so then they had this piece of rock, and then they fixed the bycatch coral with epoxy putty, and then they go with a boat, and they were throwing them to the bottom up to 100 meters, and then they were checking how, if they were about the survival, and if the colonies they were in upright position. So they call this the badminton technique, and you have more information in this reference in this publication that it's in the bottom of the slide. So we move to the coastal habitats, and here, instead of collaborating with fishermen, we collaborate with diving centers, and it was a really nice experience. And we conducted a pilot action in the Medas Island marine protected area, and we transplant more than 400 Gorgonian fragments in just one dive. So it was quite successful. And what we did, in fact, we make meetings with the diving centers. We train, we explain the method that we wanted to develop. So we went there and we took pieces of healthy colonies, and we kept them just for some hours in some coolers. We used the same epoxy putty, and then we went down diving and just fixed them all into the bottom. Okay, so and then we were checking the survival of these transplants that we did together with the diving centers. So at the beginning, the survival was quite good, but at the end of after 2 years, we saw that the survival was quite low. It was not a matter of the technical problem, because once the transplant is well fixed, there is no problem that they can stay there. But what we witnessed during this period is that we had 2 marine heat waves hitting the Catalan coast, so they kill most of the transplants by the end of the pilot action. So this is something that we already knew that they were affected by this kind of high temperature, and this is why within MERCES we conducted this transregional thermotolerance experiment that we combined with genomic and powerful genomic analysis. And the goal of this was to try to identify resistant donor populations to avoid this failure in the survival of the transplant in the different areas. So in the map, this is a sea surface temperature map, and it's just to show you that we were collecting colonies from different populations in different areas in the Mediterranean, but as well just outside of the Mediterranean from the same species. So we were working in 5 regions. We collected or sampled 12 populations, and there were more than 300 colonies. So we placed them in an experiment setup that if you want further details, I can provide it later. But the idea here is that we were putting all the populations under the same conditions, and we were checking the thermal stress across populations. And these colors correspond to the regions, the different regions that I showed you in the previous slide. And the interesting thing here is that if you see the survival rate of each population, you see that there are some areas that they behave more or less in the same way. But then we have the populations from Portugal and some populations in Italy that they behave different. So this gives some hope that there are some factors or more resistant populations to the thermal stress. Okay. So we combine, as I said, this transregional experiment. Since we know what are the response to thermal stress, then we can select the populations and the most resistant individuals, and make a whole genome analysis, and then check for the development. We are trying to identify how we can identify the resistant donor populations and individuals to be used and to be cultured in the future in the restoration actions. Okay, so this is my last slide, and this really home take message that I want to deliver is that before engaging any restoration action, we have to stop the main drivers of degradation. And of course, in the framework of climate change, this is difficult, but it's better to understand that if we don't stop the main drivers, it's going to be hard to make successful and efficient restoration actions. And for the positive, the good things that we found in MERCES is that the coral, we develop good techniques for the restoration of coral. So there's no doubt that these techniques are working. It was really good to see how this research project enhanced the collaboration with key stakeholders, and this is really important as well, because the conservation and the restoration is a matter of everyone, not the researchers, so the collaboration is key. And these techniques, they can be really useful, especially working at a local scale when we have to solve some issues at local scale. For the cons is that not all the species show the same success, so we have to check for this kind of things. The high cost if we want to go to the continental shelf, or even worse, if we want to go to the deep sea, this is a really high cost for the monitoring, etc. And probably the most important thing is that it's really difficult from the experience that we have. It's really difficult to scale up from the local scales to significant ecological scales, and this is important to know that we still need some work if we want to go to this upscale the restoration actions. And in this slide, I put the last message that because I think that is really important to say that nature has the solutions. We saw, for instance, that there are some populations that are resistant to thermal stress. So we must provide nature the best conditions to develop these solutions. And of course, restoration at local scales, for instance, can support this the resilience of the habitats and the ecosystem services that they provide. And thank you for your attention. And if you have any question, I'll be more than happy to answer it. And I just left here my email if you want to contact me. And this is the web page of our research group where Cristina Linares and others and the people that have been working on this kind of research topics, you can find more further information. Thank you very much.
Okay, thank you, Joachim, for a very good talk about all the work that you and your team have been doing. And I suppose I'll ask, first of all, a question to you, Joachim, just to clear up in case there's any doubt in people's minds. When using these various methods, and taking parts of coral that maybe have been fished up, you are then releasing these back into MPA areas. So there's no more effect on them. And probably therefore the solution that you have also requires maybe for greater areas to be turned into MPAs that then can be restored in some way. To comment on that.
Yeah, of course. I mean, the idea is that if you want to restore, for instance, for the transplants, they were placed in non-fishing areas or mainly non-fishing areas. Then there is the enforcement of the regulation, right? Because they can place the transplants in a marine protected area, but then if the area is not well enforced, the fishermen they go and fish in the area, and then all the transplants are gone, right? So yeah, the idea, as I said, we have to avoid or to be sure that the new disturbances, they will not affect the restoration areas, because otherwise it's going to be a problem.
Okay, thank you for clearing that up. So Jesper, there's a question here about, could you give an indication of the costs of the type of operation that you made? And particularly, is it possible to express it as in terms of area or the spatial scale that coral reef that might be restored?
Yes, I can. For the pilot itself, the main driving costs are the rental of the tug.
Mm-hmm.
And then, but that's of course a scaled version of what we want to do. Then if you look in the paper that we've published, I think it's called the modeling paper, we've actually gone through and discussed the costs that we foresee per larvae, and then also the next step is, in terms of a spatial scale, it's entirely dependent on the amount of larvae that you release. But we've attempted to make some indications on that in the paper.
Okay, and in terms of the number of species that you have been able to find from these slicks, are they just a few, or have you been able to have quite a variety of coral genera?
It's been quite a variety. We see a dominance of certain species. So, for example, the Acropolis species are very prolific over where we sampled, but there is a way greater range. Okay,
and there was various talks, I'm not sure quite, some of the questions seem to have disappeared on my system, about temperature and whether this would be different between the metal and the plastic incubation chambers.
Yeah.
Any issue with that at all?
Issue with that? No, I don't think there's necessarily an issue with that. Given the fact that we saw similar survival rates between the two different tanks. One thing, for example, the aquaculture systems that we had set up, we had some form of flow through on them, so we were pumping water into them, and we had big cones in the middle, essentially, through which water filtered out. Do in future, if we notice that water is becoming warmer than we would really like, with those steel tanks, we could up the flow rates of cool water in there and have essentially a more climate controlled environment. So I think it had an impact on the settlement results for the experiments. However, I think it's something we can also we can tailor a lot better in future.
Okay, thank you for that. So I'm just trying to find a question here for Joachim. What sort of, did you mention which species particularly were successful with the methods that you used? Did you have differences between the various species for survival?
Yeah, I mean, the differences were for the species that the people in Azores were working, and I have to recognize that I don't remember the names of the species, but I can, if somebody is interested, I can provide the slide that they provide me with the difference of the different species. They were working with 4 species, as you saw in the slide. And one of those had this 15% of survival rate, and the others were up to 100, and then the others were in the middle. Okay, sorry about that. Okay.
In terms of the translocation of species between sites, are there issues here that of legal rights and or socioeconomic sort of considerations of moving a species from one area to another? Do these sorts of things have to be sorted out, both in shallow and deep water? Maybe Jesper first.
Yeah, it's something that we kept in mind, because for the genetic structure, for instance, there is a these species are highly structured, so moving some individuals from one population to another can have effects on the diversity of genetic diversity that you can find. And we were also concerned about the potential risk of translocating bacterial or viruses, pathogens from one region to another. So just delivering some pathogens in a new area where the colonies or the corals or the populations are not resistant to. And we have been working on these kind of things, but it's true that the scales at what we are working, they seem at this moment, they seem that they don't have any effect on this. And regarding the pathogens, what we saw is that for our surprise, we are conducting as well this microbiome, so characterizing all the bacteria and protists associated to these coral species in the coastal areas, and it's quite similar across the 5 regions, for instance. So this gives us some hopes that translocating is not going to cause major changes, at least in this component that can cause afterwards the diseases or some kind of outbreaks on the populations.
Yeah. Jesper, have you got anything to add?
I agree with Joachim, to be honest. For the experiment we took in now, we actually released the larvae on the same location at which we got them. But I do think that there is certain discussions that need to be had before you move species from warm areas to cooler areas. That's definitely the case. Mm. And the way that we mostly also see this methodology at the moment is to say, okay, look, you have a degraded reef at a certain location, and then you try and rehabilitate a location nearby. However, I mean, it can also be expanded over larger areas.
Okay, and one other question for you, Jesper, was to do with, because I think you answered it in text, but maybe not everyone saw it. In terms of the proportion of the slicks that you find, how much do you sample, and will it have an ecological effect if you sample these slicks? Yeah,
it's a very common question we get. We've actually modeled it and basically looked at, okay, if you have a certain density of coral within an area and you assume this amount of spawn comes off it, and the amount that we harvest in our overall picture where we use the trailer suction hopper dredging, I think far below 1%. And particularly what we harvested now for the recruit pilot, if you want to call it that. I mean, we were harvesting small sections of the slick. We were still, missing a fair amount still. And not to forget, I mean, these slicks, these big streams that you saw, these are occurring all over the reef. So the amount of harvest is very, very little. Okay. And there's a question here, really. I mean, it's great looking at this bigger scale in tropical corals. Can we maybe use similar techniques for cold water corals, Joachim? Can we use AUV to collect spawning eggs? Is it going to be possible ever?
Maybe something that I had to put in the presentation, but we decided to move to the transplants techniques because since these species are so long-lived, if we have to wait, the survival rates of the larvae, they are really, really small. So we have in an area of 2 square meters that we monitor, for instance, you have 1 or 2 recruits every 2 or 3 years. So we cannot expect that the recruitment can support the resilience of this species. So we decided to move to the transplants because in that way the transplants, they have the capacity. We checked that they have if we took from healthy colonies, even pieces of I don't know 7 centimeters, for instance. You transplant them and they are already reproductive, okay? So they can then, if they grow well, they can then reproduce, and then they can provide the larvae, and then they can support the recruitment in the areas where we transplant the restoration pilot action. So this was what our strategy to develop the restoration protocols. And combine this with the knowledge that we got from the genetic studies, because we could calculate somehow what is the dispersal area or the dispersal distance that they have the larvae from the in the natural populations, and it's quite low. It's about the effective dispersion rate. It's no more than 40 centimeters away from the, or 1 meter from the parent colonies in this species. So we in the protocol, we propose that to make the patches of transplants separated by this distance of dispersal, just to enhance the natural recovery of the area. Okay.
Okay. Yeah, no, sounds very sensible. Okay, well, we've run out of time, really.
Yeah, okay.
I'd just like to maybe go to the final slide, if we can find that.
Okay, I'm going to stop sharing my screen.
Okay.
I don't know if I...
We've got to find ours. Okay, well, while that's being done, I should say that all the questions that were asked have been logged. And I will ask our speakers, since we had so many questions and quite a few of them were probably quite short answers, I'll ask our speakers if they'd like to maybe make written responses to those questions from the file that we've kept. And these will then be put on the MERCES website alongside the archive version of this webinar. And that'll be available in about, well, the webinar will be available in about 1 week, and maybe the questions a little bit longer for that.
Mm-hmm.
I should say also that for those that are interested in marine ecosystem restoration in the Mediterranean Sea, there is another blue growth project that has started a year ago called AFRIMED, and that's now approaching its 2nd year. And that also has a business club for liaising with industry and policymakers and scientists. So if you want to be kept informed about that, and it involves not just European countries, but also African nations bordering the Mediterranean and other nations, then you should keep in touch with the AFRIMED group. And if you want to, I can put you in touch with that if you send me a message at the website, at the email site that I've put up here, which is merces@deepseasolutions.co.uk, and I'll put you in touch with the relevant people. So any feedback you want to give us about the webinar is always welcome. But being our final year in the MERCES project, if you have any comments that you would like to make about the webinar series particularly, or the newsletters or the case studies, it'd be very useful to be able to reflect these back in our final reports to the Commission, which have got to be submitted by the end of March. So if you could maybe, when you shut your Zoom software, immediately send me an email. It would be very much appreciated to have your feedback that we can then put into the final report. And so thank you very much to our speakers. You did a great job. It was really interesting. You can tell by the number of questions. Thank you to GRID-Arendal for all your support for the webinar series. And thank you also to Andy for supporting me here at the NOC. So we wish you all good fortune in your efforts to restore the marine environment in the future, and long may it continue. Thank you very much, and bye-bye.
Thank you. Bye.
Yeah, thank you guys. Bye-bye.